Okay, so here we're going to talk about what happens after glycolysis. And so for glycolysis, of course, you are going to have the generation of ATP, and you're going to end up with a 3-carbon compound called pyruvate. And so you also have left behind NADH that had taken up some electrons. And so for glycolysis, what happens after depends really on the availability of oxygen. So is oxygen available? And so if oxygen is available, you will have aerobic respiration that follows. If it is not available, you will have anaerobic respiration that follows. In the case of aerobic, pyruvate will be oxidized to something called acetyl-CoA and enter the TCA cycle. NADH that was generated during glycolysis is not able to itself be transmitted across the membrane. However, it can pass its electrons to intermediates that can then be shuttled into the mitochondria so that oxygen can be the final electron acceptor. So in the case of aerobic glycolysis, you end up with 2 pyruvates from a single glucose, and you end up with your 2 NADH. So your 2 pyruvates are able to cross the mitochondria membrane. These will become acetyl-CoA, and eventually you will make ATP. For your NADH, you're going to have to pass these to a carrier, and this carrier is able to transfer the electrons to the electron transport chain, and then you can generate ATP as well. There are 2 shuttle systems. We have the glycerol 3-phosphate or the malate-aspartate shuttle systems. Now in the case of anaerobic glycolysis, you will have simply your glucose makes 2 pyruvate, and then you're going to generate 2 lactate through lactate dehydrogenase. This is important for recycling your NAD to generate more ATP during glycolysis. Now, to get your NADH electrons into your mitochondria, you're going to have to shuttle them. So the shuttle carries the NADH potential indirectly. A major shuttle is glyceraldehyde 3-phosphate shuttle. So again, we started with glucose, we did glycolysis, we generated pyruvate, and we made NADH. NADH is going to transfer to dihydroxyacetone phosphate, or DHAP, that is an intermediate of glycolysis, to glycerol 3-phosphate. And then that 3-phosphate can go through the inner mitochondria membrane, but not the, can go through the inner mitochondria membrane, and it's going to transfer the electrons to FAD that is part of the electron transport chain. And this FAD, the flavin molecule, is going to pass on those hydrogens or electrons into the electron transport chain, and then you're going to make more ATP. Now the second mechanism for shuttle is going to be a malate-aspartate shuttle. Now again, we started with glycolysis. We have glucose, we made our 2 pyruvates, we made our 2 NADHs, and our NADH can transfer to oxaloacetate the electrons, and this will generate malate. Malate is able to enter the mitochondria with a co-transport, and alpha-ketoglutarate will exit the mitochondria. Now this is all tied together because once alpha-ketoglutarate exits, alpha-ketoglutarate can regenerate oxaloacetate from aspartate. And this can keep the oxaloacetate malate shuttle going for the electrons from NADH. Now, where does aspartate come from? Well, once malate enters the mitochondria, it's going to pass those electrons to NAD and regenerate oxaloacetate. However, oxaloacetate cannot cross the membrane. whether it's in the cytosol or in the mitochondria, it's stuck wherever it's made. And so oxaloacetate via transamination with glutamate will generate aspartate, and now aspartate can leave. Glutamate can enter, and glutamate will regenerate alpha-ketoglutarate. So alpha-ketoglutarate can exit, so malate with the electrons can enter. This whole process is to get malate with those electrons from glycolysis into the mitochondria. So it can go to the electron transport chain, and then you can generate ATP. So in this malate-aspartate shuttle, what are the reactions? Well, let's just look at this a little bit. So we have transamination reactions. And basically, transamination reactions, you're moving around your nitrogens. So here we have aspartate, and you have an aspartate transaminase, and that's going to transfer the nitrogen from aspartate to glutamate. Or it's going to do the reverse, transfer it from glutamate to aspartate. And in the process, you're going to generate two other molecules. You're going to either generate oxaloacetate or alpha-ketoglutarate. Now, malate is also going to be able to become oxaloacetate through malate dehydrogenase. And this is where you get the transfer of electrons to NAD to form NADH. And then this can go to the electron transport chain, and this can make ATP. Now, on the other side, on the cytosolic side, you have oxaloacetate, and you want to transfer the NADH from glycolysis to oxaloacetate to generate malate, so malate can now cross the mitochondria membrane and transfer its electrons into NAD so it can go to the electron transport chain. And so this whole process is linked together to move the potential energy generated from glycolysis packaged up into NADH so you can get it into the mitochondrial matrix to make more energy. So these transamination reactions that we just talked about are going to be important for moving around the nitrogens and allowing the proper species to be able to be transferred back and forth. So here we have glutamate, which has a nitrogen, and that's going to be exchanged for aspartate, which has that nitrogen. And then you're going to have your malate be able to cross with alpha-ketoglutarate.